Hello everyone, we are from Group 1 in class Nurse RKI. In this change, we will present our result of experiment about carbohydrate test. Before we start our presentation, we would like to thank to our lecturer, Mr. Ahmad Farid, and now I will introduce our member. There is Diva Zahra, the Flora Balqis, and Jelviona Anissa Arvi. So, let's start to the presentation.
First is Molisch test. The purpose of Molisch test is to prove the presence of carbohydrates in a sample. The principle of Molisch test is this reaction is caused by the hydration power of concentrate inorganic acid against carbohydrates, forming furfural or its derivatives such as hydroxymethyl furfural. Molisch reaction consisting of alpha-naphthol will react with furfural to form a purple compound. And then this is the reaction of Molisch test. This is the sample for Molisch test. First, there are maltose solution, aqueous solution, fructose solution, glucose solution, sucrose solution, lactose solution, amylum solution, galactose solution. And then this is a sample for the Molisch test. There are maltose, aqueous, fructose, galactose, glucose, sucrose, lactose, amylum, and arabinose solution. We use 1% from each solution, and then the reagent that we will use and will be react with the solution is sulfuric acid and Molisch reagent with 5% alpha-naphthol solution in 95% ethanol. And then the tools that we will use is test tube, pipette measure in 1 milliliter, and drop pipette. Okay, now I will explain the procedure of Molisch test. First, enter into the test tube each 1 milliliter of sample solution. Second, add 2 drops of Molisch reagent each. Mix until homogeneous. The third, add 0.5 milliliter of concentrate of sulfuric acid to each tube by tilting the tube and passing concentrate sulfuric acid through the wall, and don't shake it. The last, a positive reaction or presence of carbohydrate is indicated by the formation of a purple ring. You can see the example of in the picture. Then the result of Molisch test is from the table of the presentation. We know that the only one solution that not contain of carbohydrate is aqueous, because the result is negative and the color of the Molisch test is clear, while the other solution from the glucose, sucrose, lactose, maltose, amylum, galactose, arabinose, and fructose solution was purple ring formed. Okay, I will tell you the discussion of Molisch test. Carbohydrate are molecule compound, polyhydroxyaldehyde or polyhydroxyl ketone. Carbohydrate exist in the tissues of plants, animal, and microorganisms in various form and level. Disaccharide have 2 monosaccharides that are linked by glycoside bond. The most common disaccharides are maltose, lactose, and sucrose. There are many carbohydrates in food ingredients. The mechanism for the formation of the purple ring is that carbohydrate by concentrate sulfuric acid will be hydrolyzed into monosaccharides. Then the monosaccharides are hydrated by sulfuric acid to become furfural. If the compound is hexose-hexose, the compound formed is hydroxymethyl furfural. To determine the presence of carbohydrate in general, the Molisch test can be used. This Molisch solution itself contains 5% alpha-naphthol in 95% alcohol of ethanol. This test is based on the reaction of carbohydrates with concentrate acid. Carbohydrates have the empirical formula and are commonly called as hydrate of carbon from Leininger 1970. In food ingredients, carbohydrate exists in several forms. Monosaccharides are commonly referred to as simple sugars, which have one aldehyde or ketone group. The furfural in the presence of alpha-naphthol will condense to form a purple complex. Pentose dehydration will produce furfural hexose hydration will produce hydromethyl furfural, while rhamnose dehydration will form methyl. Okay, finally we can get the conclusion of this Molisch test. The Molisch test is the common test for carbohydrate. Molisch reagent, which consists of alpha-naphthol in alcohol, will react with the furfural to form a purple complex compound caused by hydration power of concentrate sulfuric acid to carbohydrates. The test is not a specific test for carbohydrate, only determine the presence or absence of carbohydrate. But this test is carried out for the basis of research on what carbohydrates are contained in the sample.
And now the Benedict test. The Benedict test is a method used to determine the carbohydrate content of reducing sugar, such as all types of monosaccharide and reducing disaccharide, like maltose and lactose. And the purpose of this test is to prove the presence of carbohydrate which have property as a reducing sugar. And the principle of this test is the sugar which has free aldehyde or ketone group will reduce copper ions in an alkaline atmosphere to become insoluble and red copper oxide. And as you can see the picture below here, this is the reaction of Benedict test. And now the materials and the tools. We need 10 samples. There is maltose, dextrin, fructose, glucose, sucrose, lactose, amylum, galactose, arabinose, and glycogen solution. 1% for each solution. And reagent Benedict reaction 173 grams on NS citrate and 100 grams of Na2CO3 in 800 ml of water. And the tools, there is test tub. As a place of two solution react, you can give label the test tubs according to the names of the solution. Pipette measure and drop pipette to move the solution from one container to another, and water bath to heat the solution. And now the procedure or the step of the Benedict test. First, put in a test tub 5 drops of the test solution and 15 drops of Benedict reagent and mix it well. Since we have 10 solutions, so do it for 10 times. Second, put the test tub in water bath of boiling water for 5 minutes. Then let the test tub cool slowly. After that, pay attention to the color or sediment that is formed. Positive result have turquoise, yellow, or brick red and sediment. The color depending on the level of reducing sugar, and negative result have blue color. The level of reducing sugar you can see at the picture in the right. Blue have 0 reducing sugar, green less than 500, green precipitate 500 till 1000, yellow precipitate 1000 to 1500, orange precipitate 1500 till 2000, red brick precipitate more than 2000. This is the Benedict test result. As we can see, for amylum, glycogen, and dextrin solution with the same color result, which is blue, which means negative. Then sucrose with green color result, which means positive. And for lactose, maltose, galactose, fructose, glucose, and arabinose solution have the same color result, brick red, which means positive. And now we move to the discussion. The test result obtained from the Benedict test are in the experimental solution dextrin, it gives a green solution and a precipitate is formed, which means positive. This is because the reducing sugar content in polysaccharide is less than monosaccharide and disaccharide. And next, for starch or amylum, glycogen and sucrose produce a blue solution and there was no precipitate, which was negative. This is because sucrose does not contain free anomeric carbon atoms, because the carbon atoms of the two anomer, namely glucose and fructose, are related to each other, while starch or amylum is composed of a lot of D-glucose, fruit sugar. And then for lactose, maltose, galactose, fructose, glucose, and arabinose solution, it produces a brick red solution and brick red sediment, which means positive. This is because the result of reducing of CO2 plus ions to Cu plus ions, which precipitate as CO2O or copper oxide by an aldehyde or free ketone group contained in reducing sugar, which take place in an alkaline atmosphere. Alkaline property owned by Benedict reagent due to the compound of sodium carbonate. Next, there's conclusion. So the Benedict test is a chemical test to determine the sugar or carbohydrate content reducing. Reducing sugar include all types of monosaccharide and some disaccharides such as lactose and maltose. Benedict solution will test for the presence of aldehyde and ketone group in aldose and ketone sugar. Benedict solution contains sodium citrate, anhydrous sodium carbonate, and copper sulfate, 7H2O, and all of these salts are dissolved in water. A positive result is indicated by the formation of a green, blue, red, or yellow precipitate depending on the level of reducing sugar present. The color comes from the copper oxide compound, which is formed from the reaction.
And the last is iodine test. The principle or basis. Polysaccharides with iodine solution will form a blue or purple starch and red glycogen color complex, while oligosaccharides and monosaccharides with iodine solution don't form a color complex or colorless. The purpose: to prove the presence of carbohydrates from the polysaccharides group, and you can see the reaction in here. Next, you can see the sample, the reagents, and the tools. The sample are 1% maltose solution, 1% aqueous solution, 1% fructose solution, 1% glucose solution, 1% sucrose solution, 1% lactose solution, 1% amylum solution, 1% galactose solution, and 1% arabinose solution. Next is the reagents. Is iodine reagent, who dissolves 20 grams of Ki in 1000 ml of distilled water and add enough 12 until the solution is dark yellow. And next is the tools. The tools are test tube, pipette measure 1 ml, and drop pipette. So this is the procedure. The first step is you should enter into the test tube each 1 ml of sample solution. The second is you should add 2 or 3 drops of iodine reagent each and mix until homogeneous. Third, the solution that changes color to blue because it contains carbohydrates from the polysaccharide group. And the fourth, a solution that does not change the color because it doesn't contain carbohydrates from the polysaccharide group. And so, this is the result. You can see the sample, the iodine test result, and the conclusion. 1. Amylum solution 1%. The iodine test result is blue solution, so the conclusion is polysaccharide. 2. Maltose solution 1%. The iodine test result is not color, so the conclusion is non-polysaccharide. 3. Lactose solution 1%. The iodine test result is not color, so the conclusion is non-polysaccharide. 4. Sucrose solution 1%. The iodine test result is not color, so the conclusion is non-polysaccharide. 5. Glucose solution 1%. The iodine test result is not color, so the conclusion is non-polysaccharide. 6. Fructose solution. The iodine test result is not color, so the conclusion is non-polysaccharide. 7. Galactose solution 1%. The iodine test result is not color, so the conclusion is non-polysaccharide. 8. Arabinose solution 1%. The iodine test result is not colored, so the conclusion is non-polysaccharide. And the last is aqueous. The iodine test result is not color, so the conclusion is non-polysaccharide. And this is the discussion. The results obtained from the iodine test are in the test solution only starch solution, because starch is a polysaccharide group. Meanwhile, glucose, fructose, maltose, galactose, arabinose, sucrose, and distilled water don't change the color, which means they are not included in the polysaccharide group. So, that the color change experienced by starch indicates that starch is carbohydrate from the polysaccharide group. And the last is the conclusion. The carbohydrate content of a food ingredient can be determined by conducting a qualitative test of the iodine test method. The principle and determination of carbohydrates using the iodine test method to identify carbohydrates that contain more than 10 monosaccharides that are bound are also called polysaccharides or complex carbohydrates. These polysaccharides, when hydrolyzed, will produce glucose polymers in the form of starch or starch. The reagent used is a solution of amylum with iodine to form a blue complex. Amylopectin with iodine will give a red-purple color, while with glycogen and dextrin will form a red-brown color. This is the benefits in the nursing. The first is Molisch test. In nursing, the Molisch test is used to check the urine for which carbohydrates are contained. In patients with a diabetes mellitus, glucose is found with the formation of purple ring in the Molisch test result. The second is iodine test. To understand carbohydrate determination as learning material and information about the carbohydrate content of a food ingredient. And the last is Benedict's test. The reducing sugar test uses Benedict's solution, which can detect 0.1% glucose levels in the mixture, so it is very often used for urine and blood samples. Benedict's test can be used to detect the presence of sugar in the urine. If the urine is tested by Benedict test shows us positive result, it can be a sign of a disorder commonly called diabetes mellitus.
Thank you for watching. If you have any question, please write down the question in the comment section. See you.